And welcome everyone. Today we will discuss about the properties of immunoglobulins. Let us start. So this is a table showing all the properties of immunoglobulins. The first property discussed here is about serum concentration expressed in percentage. Immunoglobulin G is the highest in concentration among all others, followed by A, M, D and E immunoglobulins. The molecular weight as seen here, the lightest of all the immunoglobulins is the immunoglobulin G, followed by immunoglobulin A, which is 1,70,000 in monomeric form, and in dimeric form it is 3,85,000. The heaviest of all immunoglobulins is immunoglobulin M, and followed by immunoglobulin D and E. The sedimentation coefficient is the rate at which these immunoglobulins settle down, and the heavier the immunoglobulin, the greater is the sedimentation coefficient. So immunoglobulin M is seen to have the highest sedimentation coefficient. The heavy chain class present in immunoglobulins are shown here. Immunoglobulin G has gamma heavy chain, immunoglobulin A has alpha heavy chain, immunoglobulin M has mu heavy chain, immunoglobulin D has delta, and immunoglobulin E has epsilon heavy chain. The light chains are either kappa type or lambda type in each immunoglobulin. The valency of immunoglobulin G is 2, which means 2 antigens can bind to one immunoglobulin molecule. Immunoglobulin A has in monomeric form it has a valency of 2. In immunoglobulin M has a valency of 10. Immunoglobulin D and E also have a valency of 2. Number of basic 4 polypeptide chains. So each immunoglobulin has 4 polypeptide chains, the basic unit. So immunoglobulin G has is a monomer. Immunoglobulin A is a monomer in serum, and it is dimeric in secretions. Immunoglobulin G is a pentameric immunoglobulin. D and E are also monomers. So there are 3 immunoglobulins that are monomers, that is immunoglobulin G, D and E. Continuation with the properties of immunoglobulins, the placental transport is one function, and it is only immunoglobulin G which can be transported from through the placenta from mother to the fetus. The presence in milk is shown only by immunoglobulin G and A, remaining immunoglobulins are absent. The secretory immunoglobulin is immunoglobulin A, and it is secreted by the seromucous glands. Intravascular distribution is highest in of immunoglobulin M. The carbohydrate content is seen to be highest in immunoglobulin D, followed by E and M. The subclasses of various immunoglobulins are, it's only the immunoglobulin G which has 4 subclasses, G1 to G4, and immunoglobulin A has 2 subclasses, A1 to A1 and A2. The remaining immunoglobulins lack subclasses so far. Some more properties of the immunoglobulins. The complement fixation property is shown by 3 immunoglobulins, that is G, A and M. So highest complement fixation property is exhibited by immunoglobulin M. The half-life of immunoglobulins are shown here, and the longest half-life is of immunoglobulin G, followed by A, M, D and E. So the principal site of action for various immunoglobulins is shown in the next point. Immunoglobulin G and M act in the serum. Immunoglobulin A acts in secretions. Immunoglobulin D is the receptor for beta cells, and immunoglobulin E are found in mast cells. The various characteristic properties exhibited by immunoglobulins are present in this column. Immunoglobulin G exhibits precipitation, and complement fixation. It is a late antibody as it appears in the serum much late. Immunoglobulin A is found as a secretion, and it's also seen in serum. Immunoglobulin M is an early antibody. It is the first antibody to appear in the serum, and it gives protection to our body. It exhibits agglutination and opsonization. Immunoglobulin D functions are not very well known. However, they are seen in B cell activation. Immunoglobulin E is seen in anaphylactic reactions. Now we will discuss the major functions of immunoglobulins. So we'll start with immunoglobulin G. Here we will follow the mnemonic GAMDE, GAMDE. So immunoglobulin G major function is that it's a main antibody of secondary immune response. It opsonizes bacteria, making them easier to phagocytose. It fixes complements, which enhances bacterial killing. It neutralizes bacterial toxin and viruses, and it crosses the placenta. So it is the only immunoglobulin that can cross the placental barrier, and it provides immunity to the newborn. Immunoglobulin A is the secretory immunoglobulin A. It prevents attachment uh, of bacteria and virus to mucous membranes, and it does not fix complement. Immunoglobulin A is present in various secretions which include milk, mucus, etc. Immunoglobulin M is produced in primary response to an antigen. It fixes the complement. It does not cross the placental barrier as it is pentameric and it is very heavy. Uh, Antigen receptor on the surface of B cells. Immunoglobulin D is found on the surface of many B cells as well as in the serum. Immunoglobulin E mediates the hypersensitivity by causing release of mediators from mast cells and basophils upon exposure to any allergen. It defends against worm infections by causing release of enzymes from eosinophils. It does not fix the complement. The main host defends against helminth infection. So these were some of the major functions of different immunoglobulins. These are my references for the lecture. Thank you for watching. Like, comment and share and subscribe to my channel.